every lab has all of the sophisticated equipment that you would want. And even some people will not necessarily even have a lab or a conception of what technology is most appropriate for their question. They may know the science, and they certainly may know the clinical side of what they're studying, but they don't necessarily know the most appropriate technology to use to apply. And so we can help essentially with all phases of a translational study. I did bring a, oh, here's a pointer. Okay. Starting with the design of the trial and when to sample and what kinds of blood tubes to draw and things like that, to cryopreserving, banking samples, and then doing the actual assays, which can span multiple different platforms, a couple of which I'll talk about today, and then help as well with analyzing and interpreting the data, which hopefully then will inform the next set of studies. Now, in addition to that sort of one-stop shop kind of a goal, we also, as Mark alluded to, need to be aware of new technologies and be a kind of proving ground for new technologies. And having this constant stream of clinical samples and established assays that are running them is a very fertile place to try out new things and see how they compare. And then thirdly and finally, we want to be different from the average kind of core lab or the traditional kind of core lab, which gives you back your data and says, see you later. We want to actually keep that data and mine it and integrate it across studies, potentially even finding out things that were not in the original design of the studies themselves. And I'll talk a little bit as well as we go along with the story I'm going to develop in terms of the kinds of tools that we have available for integrating and mining data. So first of all, if you're going to do this one-stop shop thing and you're going to do more than 1 or 2 studies a year, you're going to have a menu of standard assays, which we have developed. Because otherwise it would be customization for every single project, and we would spend all of our time with assay development. Not to mention the fact that we then could not combine the data realistically together from one study to the next. And so among 3 sort of broad areas of cells and secreted proteins and genes, we have selected a few key assays which we've standardized. And because nobody else has talked about it so far, I'm going to take a quick digression and tell you about Cytokine, which is our most recent platform for doing these cellular assays that we had been doing by flow cytometry. And then I'll come back to the rest of this in a moment. So Cytokine is basically a version of flow cytometry in which we exchange the fluorescent labels that you typically put on antibodies in order to use them for flow cytometry. We exchange those with heavy metal tags. And the advantage of this is that now we can read these out by mass spectrometry. And why is that an advantage? Well, if you think about the fluorescence spectrum and the emission ranges of the different dyes that we have available to do conventional flow cytometry, you can see that once you get up to about 8 or 10 different colors, you have a lot of overlap, a lot of spillover to deal with, and it really becomes hard to do experiments with very many more specificities than, say, 12 or so. Whereas by using the heavy metal tags, the mass spectrometer has a very discrete readout of every single mass channel, very little nonspecific signal in neighboring channels. And furthermore, it has a really wide mass window. So the Cytokine, which is the instrument that we use, it's a commercially made machine for this, has a mass window of around 100 atomic mass units. We don't actually have 100 different elements that we can use in highly purified single isotope form, but we're up to about 40, which is considerably more than 10 or 12, right? So 40 antibodies per tube, per experimental sample, with little or no spillover. That's the real selling point of this assay. And so just to show you what we've done most recently with this is a combination of cell phenotyping and function, where we can take either whole blood or PBMCs, and I know you can't see or read the labels on all of these different dot plots, but suffice it to say that we can gate out with about 20 or so markers the various different sublineages of T cells, B cells, NK, monocytes, etc., down to the level of things like plasmacytoid dendritic cells or Tregs or other kinds of reasonably esoteric cells. I won't say that we're seeing all of the latest subpopulations that you might find in the JI, but it's a lot of well-recognized and fairly detailed lineages. Then from each of these, we're able to look at a number of different signaling molecules. And so we've got an assortment of phosphospecific antibodies to 8 different signaling nodes, which we can stimulate via a panel of different cytokines, TLR agonists, and T cell receptor, B cell receptor agonists. And by looking then at each individual cell phenotype from this list of 30 or so different cell subsets, we get a matrix for each one of them of approximately 8 stimuli times 8 or 10 different readouts, and that provides a very exquisite fingerprint of the responsiveness of that person's immune cells. What good will this do? Well, we have simpler versions of the assay that we could talk about that have shown differences in terms of studies of aging and other kinds of inflammatory responses, but we have yet to see where this more sophisticated version of the assay will take us. So I just wanted to throw that out as an example of the kinds of new assays and technologies that we're developing. But I want to turn now to spend the rest of the time talking about something a little bit simpler, which is actually secreted cytokines, which we measure by an immunoassay. And we are actually just moving in the core, thanks to some hard work by Yael Rosenberg-Hassan, who's our technical director, to a 61-plex assay, which means we can measure in a single small sample of less than 50 microliters 61 different human cytokines. That could be a serum sample or plasma, or it could be something like a stimulated cell culture supernatant. And the way this technology works, it's actually pretty proven technology now. It's been around for at least 10 or 12 years, but it has expanded over the years. And so what you have is a set of hard-dyed beads, and the typical system that we use uses 2 different dyes which are mixed with differing intensities to make this grid or this matrix of up to 100 different bead addresses, each one with a different combination of dye. And then each of these can separately be labeled with a capture antibody for a particular cytokine. Now that's all done for us actually by the companies that we collaborate with, and we buy then a kit which has all of these capture antibodies on beads all ready to go, and we simply add our sample, followed by, I'm missing a little step here, followed by a detector antibody which can be bound to a fluorescent molecule, PE. And so then this is run through a simplified version of a flow cytometer, which essentially detects 3 different colors. The 2 colors associated with the bead address, and the 3rd color associated with the amount of bound detector antibody. And so thereby you get a quantitative readout of all of the different cytokines that you're measuring in a single small sample. And it's a kind of a barometer on the circulating inflammatory condition that a particular person is exposed to. Obviously, the blood or the serum is not the place where all immune responses take place, but it's a bit of a clue to what might be going on elsewhere. So if we turn now to the third point about integrating and keeping the data, we can use the data from this Luminex assay as well as the CyTOF assay, as I showed you just a moment ago, and other kinds of assays that we do. And we can upload all of the reports from these assays that we normally generate anyway in the course of analyzing the data. And we can upload them to a database which is online and which provides flexibility in terms of the way you can basically aggregate and read out these data. And so in this particular view here, we've chosen a particular project which was headed by Gary Fathman. It was an aging study in which we looked at a series of different immune parameters, including Luminex, on about 250 healthy aging adults. And so this is a kind of an event chart where you can see the various assays that were done and the breakdown by gender, female and male. I should say sex, but so the database people don't always get these things right. And so you can see here the number of data points that we have on males and females. What you can do in this system is, in addition to choosing what readouts you want to put on the rows and columns, is you can, for example, expand this CyTOF line and then see all of the underlying analytes within that assay so that you can see the readouts averaged across the samples for every single analyte. And similarly, you could double click on one of these cells and see the individual data points that make up that average. So there's a lot of different flexibility and ways to view the data here. And so I'm going to use this particular study as an example to then delve in a little bit further. One of the things you could do with an extract, if you drill through on all of that data and bring with it the male and female annotation, is you can basically do a heat map, and you can look at the 51 different cytokines. There's actually 54 rows here because we include some control beads which are not actually measuring cytokines, but they're measuring a kind of a quality control of the assay. And on the left side here, you've got all the females, on the right side, all the males. And just looking at the patterns as they're laid out here with the cytokines in alphabetical order, there's no huge overriding difference that you can see between males and females. There is some heterogeneity, but there's heterogeneity in both males and females. What we can do, of course, is we can cluster these. And if you cluster both the cytokines, now you can start seeing some patterns of cytokines that are highly expressed, intermediately expressed, low expression levels. But when you look at the clustering of the samples, and I know you can't read it, but suffice it to say that the males and females are pretty much randomly intermixed here, which is a clue that sex is not the driving force for the differences between these samples in terms of their cytokine levels. But it's going to get a little bit more interesting than that. So one of the tools that we have available in our Stanford Data Miner, which is this online database I showed you, is a visualization tool called Parallel Coordinates. And I should mention that both the database and this tool were developed for us by a collaborator, Janet Siebert at Cytoanalytics. So what Parallel Coordinates does is allow you a kind of visual way to look at the relatedness of different variables within your data set. And so in this particular example, we've colored by females and males, and the program actually has picked for us the top 10 analytes in the Luminex assay by CV. So by the spread of the data within this particular study, these are the top analytes. They're the ones that have the most variability between samples. Now, most of these do not actually have a sex bias. An exception is leptin, which is significantly higher in females, and PDGF-beta-beta, which is significantly higher in males. That's been published. The rest of them don't really have statistical significance, but there is something interesting that I didn't realize until I plotted this data this way, and that is that the outliers, the high outliers, for all except for this one, all of the highest CV analytes, again, chosen by the tool and not by me, all of the outliers are females. I guess there's one exception here, a male. All of them are females. If we go further and we highlight only the top outliers for one of these analytes, interferon beta, we see that those same outlier individuals are often high for other analytes as well. So they're multiple hits. They're outliers for multiple analytes. And they're all females. So I think this is just a clue to the kinds of things that we can find when we start mining data in a large human population where there may not be statistically significant differences at a group level, but there's definitely a clue here about individual patterns and individual variability, which is somehow related to sex. There's another tool that Janet has developed, which is going to be in the Stanford Data Miner very soon, and that's a decision tree, which is a kind of machine learning tool. And so taking, again, this exact same data set from this aging study, and only the cytokine data now, Again, I could do more advanced things and add all of the assay data together, but for purposes of this presentation, I'm just going to talk about cytokines. And you can ask this algorithm to see if you can predict the sex of the sample based on the cytokine levels. And so the algorithm will look for breakpoints in any of the analytes that have significant difference, that have some breakdown between the sexes, and it will essentially create a tree, a decision tree, whereby if it's above a certain level for leptin, you assign it as female, and then you go to the next analyte, and above or below a certain level, you divide again. And then, you see how well you are able to predict the sexes. And it turns out that there's two things that are kind of funny about this result. One is that this analyte is one of our QC beads. It's not a cytokine. So what is that telling us? It's telling us that none of the cytokines really are very good at driving differences between the sexes after leptin, okay? So that's one thing. And the second thing is that these decisions that it made are not all that good. So there are a lot of misclassified people as a result of this decision tree, again indicating that at a group level, it's very hard to differentiate males and females simply by their cytokine patterns. So it's really an individualized story and not a group story. And so I want to finish with this idea that, since I came to the HIMC, we thought that we wanted to be able to derive some normal ranges for the kinds of complex assays that we're doing. And I've realized over time, and talking with some of the smart people in Mark's lab that have been looking at these data over the years as well, that that's really hard. One, because the assays are not necessarily exactly stable over time, but also because of this phenomenon of kind of individualized patterns of expression. And so if you think about the serum cytokine data, there's a subset of individuals that have elevated levels of several cytokines in parallel. You saw it a little bit in the heat maps, but it was most apparent when you started looking at the parallel coordinates views. And that implies then that some of the readouts in this data are only meaningful in the context of other readouts. In other words, you might be within the normal range for cytokine X, but only if you also are elevated for cytokine Y and maybe Z, okay? Whereas that same level of cytokine X, when you're not elevated for Y and Z, might be abnormally high. So normal ranges can't really be approached in the typical diagnostic way that people have thought about them before for these kinds of complex immunological readouts, because the immune system is a balance. It is a dynamic system that when you stretch one part, you stretch another. And you could hypothesize a lot of these kinds of relationships based on biological knowledge. The percentage of Tregs that you have might need to be balanced by the percentage of effector T cells. IL-10 versus interferon gamma, pro versus anti-inflammatory, and I'm sure you could think of lots more. So again, a normal range probably needs to be substituted for a pattern, or as Jake Glanville, who's helping us work with some of these data going forward, calls it an immunotype, where you can place an individual into one of several potential patterns and thereby know whether they're normal or not normal, and maybe eventually from those immunotypes even prognosticate what kinds of other events immunologically they might be at risk for or prone to. And so I'm going to basically stop there and just sort of summarize that the immune system is highly personalized. And so we hear a lot about personalized medicine, and I think this is a personalized medicine story. But it's not just things like age and sex. There may be many other factors that are driving these sort of individualized immunotypes, and therefore the ranges, the idea of normal ranges will not be as simple as what we've seen compared to something like, say, a cholesterol test or other kinds of serum chemistry markers, let's say, where you actually have quite well-defined normal ranges, and when you go outside of those ranges, you're sick, okay? And it doesn't matter who you are, you're sick. You could be male, you could be female, but you're sick when you get out of that range. And here I think it's going to be much more individualized and much more pattern associated. And so we need to gather sufficiently comprehensive information. And I think the good part of this is that it's validating to the approach that we've been taking and that Mark has been advocating with regard to systems level studies and the HIMC in particular, because you may have a hypothesis about some set of pro-inflammatory cytokines that you think is associated with the disease that you're interested in and that you're studying. However, you're going to come to the HIMC and we're going to say, we will run 61 cytokines. And many people come back and say, but I really am only interested in these 6. And we'll say, well, we'll run 61. The other, 50 some are for free. And oftentimes then they find that there are a number of surprises in that data. And the other analytes are in fact quite interesting and maybe even, as I alluded to, necessary to inform the information from the first set of analytes that they thought were primarily of interest. And then, just to finish, this is scratching the surface, right, of the kinds of data we have. I've showed you one assay, a Luminex assay, from one study out of several different assays and a large number of studies, even if you consider just the biggest studies in the HIMC. And while the investigators that we're doing these studies for are certainly analyzing their data in some way, there is a huge room for us to do further and more comprehensive analysis and to apply new analytical tools to try to make sense out of these data and come up with ideas like immunotypes and what they might be prognostic for. So I'll stop there and just mention that we've got a big group of people now working in the HIMC, both on the research side and in the core lab doing service work for other investigators, as well as analyzing data and so on. And Yael especially is our technical director and head of the immunoassay group. So a lot of the data I showed is thanks to her. And again, the study I showed was conceived by Gary Fathman, who along with Mark were initiators of ITI and the HIMC, and the database, again, by Cytoanalytics, Janet and Wes. So thanks for your attention. I'm happy to take questions.
I think, is it on? It says it's on, but I don't.
I can kind of hear you.
Okay, yeah. Okay. So this is really interesting, and is kind of a philosophical question about what is normal. And so, looking at your outliers, I'll use a personal example. When I was a postdoc, asked to serve as a quote-unquote normal for a big cytokine study, and I was an outlier. I was one of those people, and then. And in particular with IL-12, I was a huge outlier. 2 years later, I got diagnosed with Graves' disease, and really high IL-12 is, kind of one of the characteristics. So prior to that, I was considered a normal. But maybe those outlier responses that the other postdoc blew off, as she joked that I couldn't be included in her study, so I guess I'm wondering, to what extent can some of this be predictive? When you're looking at kind of those individuals.
Yeah, so we certainly would hope that it will be, right? And I think that that's exactly where we'd like to be, is to be able to use those data and say that, yeah, you're at risk for this or that because of these profiles. Yeah, Mark. You want to?
Sure. Right, and so that's why we need longitudinal studies, and I'll talk about the longitudinal study that we're doing with Corey and with Holden that's now in its, 8th year maybe, coming up. So exactly, that's definitely one of the things. But what I was going to comment, because I hadn't seen some of this data before, is to say it jives with the thinking that there are a series of immune states, or there's some hopefully not incredibly large number of different immune states, and you could think about, and we see other kinds of evidence for this, inflammatory states, and especially in older people. But sepsis could be considered one state. The phenomena of children in developing countries that are resistant to vaccine infection and probably have a tremendous burden of infectious disease could be another state. So I think even just defining these states, because we basically have to do that, and that could be predictive, and that could help to define what normality is, or you could bin people hopefully in these different states and see what happens to them later on, if anything.
So the question I have goes back to the sex difference, where leptin pops out, and leptin is, of course, a adipose tissue hormone. So women have more fat. Do you adjust the data for confounders so that you can actually tease apart things that really don't relate to sex per se, but relate to the fact that one of the sex differences is adipose tissue?
It's a good question. I'm not sure how we would do that, but measure BMI perhaps. Yeah, it's. You want to comment? Great. I like it when people will answer the question.
I'm going to show later on that we observe these differences in leptin as well, and we have demographic data that basically have BMI and also the age of the individual. We have adjusted for that actually, and we found the differences not only in leptin but also analytes, and that we'll show later. I'm assuming those differences between the Ellison cohort that Mark will present in just a bit are due to the age differences as well. We have a broader age range, and most of the differences we're seeing are in young individuals actually, and not too much in the elderly.
It would be interesting to build, and maybe it's built into your models and your algorithms, a permutational analysis so that you have an ability to establish these normal ranges based on inter-assay and inter-assay variation. So doing a permutation, it would be interesting to see what would happen when you do a permutational analysis of the data set that you examined, and see whether or not there's a threshold where those normal ranges might be broader or might be narrower.
Yeah, that's a good point. And I do think that we're hoping that there are enough consistent patterns in the data that that everybody isn't their own individual, right? I mean, to some extent they're going to be, right? We want to be personalized in personalized medicine, but we don't want to be too personalized. Otherwise, if everybody is different, then there's no real predictive power in a group. So we hope that we can still preserve that.